All right, guys. Well, welcome to the TBL Day on the stomach and the small intestine and the digestive processes within those two organs. So let's get going. Well, first of all, actually, this background you see is just kind of a cool image of what the microvilli and villi in the small intestine would look like. All right, let's start with the stomach. So the stomach, the anatomy of the stomach is made up of multiple mucosa. And there's different layers in the stomach, and the mucosa of the stomach is mostly simple columnar epithelial tissue, which contains a bunch of goblet cells that produce and release a bicarbonate-rich mucus. And the bicarbonate-rich mucus, that would be alkaline. All right, so that would go against the strong acid from the stomach to just protect those cells a little bit, just on the lining of the stomach, and the acid could be out in the middle, but the alkaline substance would protect the cells on the lining of the stomach. But even with that, those cells still die and get turned over every 3 to 6 days. Another feature of the stomach are the gastric rugae. These are longitudinal folds in the stomach. And so it's all folded up when it's smashed down and when it's empty, but then those are what allow it to expand. Can go from 50 milliliters, which is pretty tiny, up to 4 liters, right? Like the size of two 2-liter soda bottles, right? Then there's the gastric pits, which are the site of exocrine and endocrine secretions. And in those pits, we have 3 main cell types that are important to know. So the parietal cells, these secrete hydrochloric acid. Parietal cells, hydrochloric acid. Chief cells, these secrete zymogen, called pepsinogen, which is important for breaking down protein. So that's some chemical digestion of protein. And then there's enteroendocrine cells, which secrete gastrin into the blood. So that's important. Those ones secrete into the blood, whereas both of these are exocrine. All right, this one's endocrine because it goes into the blood. The pyloric sphincter is the gateway between the stomach and the beginning of the small intestine, which is called the duodenum. Okay, lots and lots of juice gets secreted by all these, mainly those mucus cells, but to the tune of about 3 liters a day. So it's a lot of fluid. Stomach acid is strong enough and has a low enough pH to actually dissolve a nail if it's in there long enough. We'll show a video. You'll see a video during the TBL day that shows it with a McDonald's hamburger. It's pretty gross, but we'll check it out. Okay, so this is just the anatomy here. The question there, which are the cells that are endocrine? So really, if we're looking here, we're zooming in there to one section here, right? And then we're zooming in a little bit closer from there to this little section right here, right? And over here, this is really where we see most of the cells. So A is just going to be those gastric pits. B, we're going to see there, here, and down here. Those are the parietal cells. Okay, and then again, the parietal cells are the ones releasing hydrochloric acid. Okay, C are those purple-looking cells. They look like they're releasing something into the gastric pit as well, so those would be exocrine. So those would be our chief cells, releasing pepsinogen, right? And then the hydrochloric acid actually converts that into pepsin, which is the active form of that enzyme that'll break down the protein. And then we've got D, which are those enteroendocrine cells. You can see little vesicles going this direction out into the blood instead of this direction like these are, releasing things into the gastric pit. And then over here, E is that sphincter, right? The big pyloric sphincter right there. And then F is just those gastric rugae, those folds that allow for expansion. And you can see very clearly that there's multiple layers of muscle in the stomach, all going different directions. That really helps it to smash down and push things around. Okay, so the stimulation of digestion begins with just thinking about smelling or tasting food. right? That also stretching out the stomach does this, and also the presence of partially digested proteins will stimulate gastrin release, but these things here stimulate the parasympathetic nervous system, which stimulates those glands to release all that stuff we were just talking about. Okay, so gastrin, that hormone released by those enteroendocrine cells, does multiple things. It stimulates the chief cells to do their job of release pepsinogen, stimulates those parietal cells to do their job of releasing hydrochloric acid, which has some negative feedback when the pH nears 1. Okay, it gets all the way down to 1, and then that tells the parietal cells to stop releasing hydrochloric acid. And then gastrin also stimulates smooth muscle contractions of the stomach and the whole digestive tract. Chyme is a term that is used to just denote a mix of those gastric secretions and the partially digested food. And proteins are really the only things that are the only nutrient digested here, and it's done in a couple ways. It's done by denaturation, which is from the low pH, it makes those proteins start unfolding, and then pepsinogen does its job when it gets turned into pepsin to break down proteins into smaller peptide chains. Interesting thing about caffeine, real common that people have their morning cup of coffee, and that stimulates them to go take their morning poop, right? And there's a reason for that. It's because caffeine is a direct stimulator of gastrin release. That causes those smooth muscle contractions, right? Gets everything moving. Okay, one pathology in the stomach, very common in our country, is a stomach ulcer. And we used to think it was all caused by things we put in our stomach, like medication, spicy food, acidic food. But now we know that 95% of them are caused by bacteria. And those bacteria are shown here. They literally just eat their way through the stomach lining and cause big holes in the stomach like this. They just burrow right through the mucus layer and the epithelial tissue. So most of those are treated now with antibiotics instead of something else. So the absorption and timing of the stomach here, what nutrients are actually absorbed here, meaning pulled out of the digestive tract into the blood, really only a couple: alcohol, and then some drugs like aspirin, okay, and ibuprofen would be another example. And so the time course, a liquid meal leaves your stomach in about 1 to 2 hours, whereas a solid meal takes about 2 to 3 hours just to get out of your stomach, okay, and then that's just beginning the process of the intestinal tract. Okay, a couple things about how the stomach works to churn up and stir around food. Peristalsis, contractions push the chyme forward, okay? So they push it forward, as it shows here. So these contractions are pushing down this way. That pushes everything here, lets a little bit out of the sphincter, but then that sphincter pretty quickly closes. So then most of the, sorry about that. All right, coming back. There we go. So most of the chyme smashes up against that sphincter and kind of churns back, comes back this way, gets stirred up, and then it pushes down again, right, smashes a little bit more, comes out, but then a lot of it smashes and comes back. And so it's kind of this constant churning process that the stomach does, really mix up the food, digest it, and get it ready for the next step in the small intestine. Okay, so moving on to the small intestine. The small intestine is made up of 3 sections: the duodenum, the jejunum, and the ileum. And so duodenum is tiny, only 10 inches. Jejunum is huge, 8 feet long. Ileum's even longer than that, 12 feet long. Right, so we've got about 21 feet of small intestine inside of us, incredibly long. Right, all folded up all over the place, and those are in the order that it goes. Okay, so we have 2 different types of kind of folds in the small intestine. The circular folds are deep folds in the mucosa and submucosa, up to a full centimeter deep, and the function of those is actually to slow things down. So instead of folds going straight down longitudinally, they're going around, so it actually slows things down instead of helps it move, and that's what we want. We want more time for diffusion, more time to absorb the food out. We have villi in our small intestine. They're little finger-like projections, and that's what we see on the back or on the background here. These have capillaries inside of them, as well as lacteals, which are part of the lymphatic system. Both these are important for absorbing nutrients. And then we have microvilli, which are just little tiny finger-like projections on top of those villi. Okay, these are extensions off of the simple columnar epithelial tissue, and this is really what makes up the brush border. We're going to talk about the brush border enzymes here in a sec. So this is what the small intestine looks like, right? The little duodenum, jejunum, and ileum. Okay, so the small intestine, we see it here. We can see those circular folds. We can see how they're not going with the direction of food flow. They're going opposite of that, so it slows things down. B, we see those villi, so those are the big finger-like projections sticking up. But then if we zoom in on one villi there, then we're going to see a bunch of little tiny, well, there's the villus first, which will be one villi, right, which is called a villus. And then we zoom in on that, and we see microvilli. Okay, so there's the one villus. Zoom in on that over here, and we see all those little tiny microvilli on top. And this is at the cellular level, obviously. And then if we look inside of one of those villi, then we see that there's capillaries in there, and we see the green one is the lacteal, which is part of the lymphatic system, which is important for absorbing fat. Okay, another very important organ for the digestive system is the liver. The liver has 4 separate lobes, and the cells inside of the liver are called hepatocytes. Anytime you see hepat or hepato, we're talking about the liver. Okay, these are arranged in lobules, as shown here. Okay, so there's a cell in a little lobule. It's got a central artery, and we'll see that in a sec. Okay, this is another unique setup as far as blood flow goes. This is another portal system, right? Like we talked about with the hypothalamus. The hepatic artery is delivering the oxygen-rich blood. This is what gives the hepatocytes their oxygen. Okay, but then the hepatic portal vein is delivering blood from the small intestine. So this delivers nutrients for processing and storage. Okay, so that's unique that we would have a vein delivering blood to an organ instead of an artery delivering blood to an organ. But obviously we have the artery part here, but then we have the vein part here, which is the unique part. Okay, so hepatocytes are in direct contact with the blood because it seeps through these very leaky fenestrated capillaries. This is what allows it to absorb nutrients. And then the blood comes from the artery and from the vein, all empty into a central vein, and then all of those central veins that we see here, they all dump into the hepatic vein. And so this is what that looks like. Right, so here's a lobule, okay, and you can see there's a bunch of hepatocytes within a lobule. Right, there's a central vein here. Okay, we can see that we have the red ones, which are the hepatic artery right here. Okay, those are going to give oxygenated blood to those hepatocytes, and then we have there's the hepatic arterial, the red one. The purple one is the portal venual, so this is connected from that portal vein. That's delivering blood from the digestive tract, so those cells can absorb all of the nutrients. And then this one is a bile duct. We'll talk about that later, but the liver helps us make bile, and that's actually going to be sending the fluid back the other direction towards the small intestine. Okay, and so we can see that with each of those, and then these two are going to dump into this central vein, which this is finally going to make its way back to the heart to recirculate the blood. Okay, so the functions of the liver: makes and secretes bile into the small intestine, about 1,200 mils per day, somewhere between 250 and 1,200 mils per day, mostly of bile salts. Okay, so these salts are derived from cholesterol. Their function is to help us deal with fats, okay, emulsify fats. And what it means by emulsify is think of any time you've gotten some butter on your fingers, or maybe even some oil on your fingers, and you just try to run it under water and it doesn't do anything, right? It just stays on there. But if you put a little bit of dish soap on your hand, then it immediately breaks that up. And that's an emulsifier too. That's a detergent that pretty much gets between the fat molecules and breaks them apart so they don't create that film over your fingers. And that's exactly what bile salts do as well. They get between those fat molecules, break them up, make them into smaller fat molecules. We'll talk about that in a sec. So and then there's biopigments that also are included in there, along with the salts. These contain something called bilirubin, which is the product of breaking down red blood cells, and it gives the bile its yellowish color. Talk more about that next time as well. So the liver is also very important for making plasma proteins. We'll talk about a lot more of those when we talk about the blood. And then the liver is important for blood detoxification. And then the storage of nutrients. The liver is our second largest site of glycogen storage. Vitamin A and iron are also stored there. So 2 other organs that are important for digestion are the gallbladder and the pancreas. Okay, so the gallbladder is a small sac. It's really just like a little bag with smooth muscle around it, and that's connected directly to the liver and the duodenum. So its job is just mainly to store as well as to concentrate bile, and then when it's stimulated to do so, it secretes that bile into the duodenum via the bile duct. And then the pancreas. Now again, we're only talking about the exocrine portion here. We've already talked about the endocrine portion some. We'll talk about it more when we talk about diabetes. But the exocrine portion is the portion that secretes things into the digestive tract. And that's driven mainly by cells called the Acini cells. Those secrete what's called pancreatic juice into the duodenum. The juice is made up of a bunch of digestive enzymes and a bunch of bicarbonate, the high pH, because we need to counteract the low pH that's coming out of the stomach. So this is the duodenum and the organs around it, right? So we've got, A, that little green bag that's connected to this bile duct here, that would be the gallbladder, right? B, we can see it's part of the small intestine, that's the duodenum. C is the end of the duodenum, starting of the jejunum, and D is the pancreas, right? So it's all kind of collected right there in that first part of the small intestine. We've got those 3 other organs right there as well. Okay, so a little bit more physiology on the hormones here. Hormonal control of digestion. 2 important hormones we'll talk about here are secretin and cholecystokinin, or also known as CCK. So secretin is released by the enteroendocrine cells of the small intestine. The stimulus for it to be released is chyme entering the small intestine. Okay, it has 3 main actions. It stops those parietal and chief cells from releasing their things, because the idea is that now the chyme's out of the stomach into the small intestine, so we don't need the stomach to keep working. Now it's time to turn on the duodenum. Okay, it also, and same idea, it decreases stomach motility, so it slows down the chyme entering the small intestine. So if we've got enough chyme in there already, it says, let's wait a second till we get any more. Okay, and then it also stimulates bile production and pancreatic juice production and release. Okay, so pancreatic juice production and release, but bile production only. Okay, and then CCK, the release and stimulus are the exact same as above, so that's easy. One less thing to memorize, but the actions are different. The actions are, this is what causes the gallbladder to contract, causes pancreatic enzyme secretion, and satiation. Makes you feel full, like you're not hungry anymore, right? So let's take a look at this on this hormone loop chart here. Okay, so you recognize the organs. We'll start over at number 1. Number 1, we see chyme entering the duodenum. All right, so it's coming from the stomach into the duodenum. Okay, that's immediately going to cause a couple things, right? We see some enteroendocrine cells here. That's going to cause them to release secretin and CCK. Secretin and CCK are going to get into the blood and do a couple things. They're going to stimulate pancreatic juice release. Okay, and then number 4, we see those hormones make their way up here. Some of them go right to the pancreas and cause that pancreas to release the pancreatic juice. We see it right there. Okay, and then we see those hormones still up in the blood. Now they're going up to the liver. Right, Number 4 is they stimulate, secretin specifically stimulates the liver to produce and release bile. But then number 5, we see some actions there on the gallbladder. That would be the job of CCK to stimulate gallbladder contraction and sphincter relaxation in the gallbladder, which is what, or in these little ducts, which is what allows it to go down. Okay, so definitely important to know all of that. All right, now we're going to talk briefly about how each macronutrient is digested and absorbed. Okay, so we'll start with carbohydrates. Carbohydrates start all the way up in the mouth. We talked about that already with the salivary amylase, but once we get down to the small intestine, that's where the next part of it happens. We have pancreatic amylase. Okay, this is released by the pancreas in response to CCK and secretin. Breaks down polysaccharides, so those long chains of monosaccharides into. Uh-oh. iPad's freezing up there. It'll probably catch up in a sec. So it breaks down those polysaccharides into disaccharides. Okay, those disaccharides, again, that's 2 monosaccharides together. And then finally, we get little tiny brush border enzymes that are found here in the brush border to break down those disaccharides into monosaccharides. So that's the digestion part, and now the absorption part. Now that we've got them into monosaccharides, they get absorbed through the intestinal cells into the blood via co-transport with sodium. Okay, that's going to be a common theme, right? So we can see that here. We see glucose coming in, we see sodium, we see this little protein that transports both of them at the same time, right? So we remember that sodium is always higher outside of the cells, right? So that means we have a concentration gradient to push sodium in, okay? We may not have a concentration gradient for glucose, just depending on the situation, but the sodium concentration gradient helps to pull that glucose in as well. And then, in order to maintain that gradient, we have to keep the sodium, we have to get the sodium out, right? So the sodium has to get pushed out. That happens via the sodium-potassium pump we talked about last semester. This requires some ATP, so that pumps that sodium out back into the blood, so we maintain that gradient, and we can see the glucose and other monosaccharides getting transported into the blood there. Okay, so the sodium-potassium pumps help by maintaining the gradient for sodium. Okay, so why does Gatorade have both sodium and sugar in it? Well, there's a couple reasons. We need the sodium. We need both of them. We need the glucose for energy. We need the sodium for electrolyte balance. It's important to have them together because of this right here, because of these co-transporters. Right? We need, by transporting, by having glucose and sodium together, that the presence of glucose helps the sodium come in, the presence of sodium helps the glucose come in. And that's one of the reasons why there's both. Obviously, the other reason is that nobody would drink just salt water, right? It would taste terrible. So, all right, so that's carbohydrates. What about proteins? We have what are called pancreatic proteases. There's 3 there on the right, and you can see the inactive versus the active form. Okay, so trypsinogen gets activated into trypsin, chymotrypsinogen into chymotrypsin, procarboxypeptidase goes into carboxypeptidase. These are all the active forms over here, inactive there. Okay, so these proteases break down long peptides, which means a lot of amino acids strung together, into smaller peptides with just a few amino acids. But then finally we got to get down to those brush border enzymes again, so proteolytic enzymes. Lytic means to lyse, to break down. Proteo means protein, right? So these little enzymes finally break them down into individual amino acids, and that's what we need to get them in. We need to get them down to individual amino acids before we can get them into those intestinal cells. So that's the digestion part. The absorption part into the cells is via co-transport with, you guessed it, sodium again. Same thing. Okay, this is what it looks like. We go from long polypeptide chains all the way down into single amino acids. Then we got co-transport with sodium, just like before. Still, that sodium-potassium pump is important to create that gradient. All right, and then it shows the amino acid getting into the blood. Okay, finally, lipid digestion and absorption. So gastric lipase there is some of that present in the stomach, and I know I just mentioned before that proteins are the only thing that's majorly digested in the stomach, but there is a little bit of fat digestion in the stomach by this gastric lipase. It starts to break up lipids a little bit. Then those bile salts we talked about that emulsify fats like a detergent, surround them and separate them from others. And then pancreatic lipase breaks down those chunks of fat into smaller fat droplets. And now we have to use a special carrier that's provided by the pancreas as well. And these are called micelles, which are pretty much allow the polar side of the fats to face out towards the water. And that allows those fat droplets to stay small, and then that helps carry them to the brush border enzymes. And then obviously the micelles were formed. Micelles deliver fatty acids and monoglycerides to those intestinal cells, and then that dumps them into the intestinal cells. And then we see inside the intestinal cells, they get processed some more. They get surrounded by a protein, and that turns them into something called chylomicrons. Those chylomicrons are then dumped out of the intestinal cells into the lacteals, which again are part of the lymphatic system. There's like lymph vessels, okay, and then those are eventually going to get dumped into the veins as chylomicrons. And then eventually those veins are going to dump back through the portal vein to the liver, and that's when we finally convert those chylomicrons into something we're more familiar with, cholesterol, right? Low density lipoprotein. That's what LDL is. That's your bad cholesterol. And then HDL is just the same, but high density lipoprotein. That's your good cholesterol. Okay, this is kind of what it looks like here. I'll let you guys go ahead and take some time and fill that stuff in, but definitely study this well. Okay, this gets us to step 4, which gets to the micelle formation. Step 5 and 6 are shown here. Those micelles get up to the brush border, dump those lipids into the intestinal cells, and then we can see those intestinal cells create this chylomicron by covering them with proteins, and then that chylomicron gets dumped into the lacteal, and then finally that chylomicron is coming from the intestine here into the capillary, through the capillary to the liver, where the liver converts it into these different types of lipoproteins, or also known as cholesterols. All right, guys, that's a long one. That's it. Study this hard. We'll see you on Monday for the TBL quiz.